All right, so when we are looking at eukaryotic gene expression, of course, I think it's going to be useful to go back again and review prokaryotic gene expression. And this is where it gets a little bit dicey. So as a reminder, eukaryotic genes don't have operons. Eukaryotic genes do not use operons. They are only going to be found in prokaryotic genes. So as a review, operons are going to be stretches of DNA, and they're going to have a whole bunch of stuff in them. The operator, the promoter, and then a whole bunch of genes. Maybe one, but usually like 6, 7, whole bunch of genes that have a similar function. The operator is going to work as a switch, basically acting as an on or off. The promoter is where the RNA polymerase attaches. So if the operator is on, then the promoter can attach. If the operator is off, then the promoter cannot attach to the RNA polymerase. So far, so good? Okay. And as I said, those genes are going to be like a whole sequence of genes. So just starting the sequence is going to lead to the transcription and translation of all of those genes. All right. Review here. We can have two different ways of controlling operators. We can either use repressors or activators. When we talk about repressors, we're thinking about negative gene control. And when we talk about our activators, we are talking about positive gene control. Okay, so the repressor works to turn the operon off. Activators turn the operon on. So many double negatives, I apologize. And when we're looking at negative gene control, the two that we are thinking about are going to be repressible and inducible. Raise your hand quick if you remember most of this stuff. Raise your hand if you're like, yeah, I could review this and I'd be okay. And raise your hand if you're like, oof. All right, not major oofage, that's good. So quick review here. Here is the trip operon. Remember that this is going to be a repressible. And the reason that it is repressible is that tryptophan acts as a co-repressor. If tryptophan is present, then it activates the repressor, which represses the gene and prevents transcription. Why would you do that? Well, if you already have tryptophan, then it doesn't make sense to make more of the genes that make tryptophan. If you've got it, don't waste energy making more of it. In terms of if you don't have it, then your repressor stays inactive. That's going to allow the polymerase to bind to the operator, promoter, go, genes are expressed, life is good. Questions there? Again, we're only thinking prokaryotes right now. Similar to that, we have the lac operon. This is inducible. It is inducible because the presence of lactose. Remember that lactose and allolactose are isomers, so if you have one, you're going to have the other. The presence of lactose causes the repressor to be inactive, so we've offed our off switch. Double negative there. I know, it's a lot. But now our repressor is inactive, it's not working. That means the polymerase can bind, can transcribe, genes are made. Voila. If you don't have lactose present, it doesn't make sense to make the enzymes that break down lactose, and so the repressor is going to stay active, turn the operator off, prevent transcription. Raise your hand if you are recalling all of this. Wow, I like it. Okay. On the flip side, positive gene control, that is when we're looking at the relationship between glucose. And so if you have glucose, what do you also have a lot of? A lot of ATP. And if you have a lot of ATP, what do you not have much of? CAMP. Remember, cyclic AMP is going to be kind of that really, really, really empty battery. So if you have low glucose, you have low ATP, you have high CAMP, and if you have a lot of CAMP, CAMP is going to act to activate, do I have it there? No, I have it up here, sorry. Doop, doop, doop. CAMP is going to act to activate the activator. That is going to promote RNA binding to the promoter and allow for transcription to occur. So these pictures are kind of opposite of what I just drew out. I apologize. Arrows all over the place. Good luck deciphering. Okay. Give me a 5, 4, 3, 2, 1 with where you would be at with a little bit of studying. Like, I know that most of you probably couldn't take the quiz on it right now. Okay. All right. So now we need to look at eukaryotic genes because things are going to be a lot different. Eukaryotic genes are way bigger because eukaryotes are complex. So the fact that eukaryotic genes are bigger has a whole host of consequences. Each chromosome is going to be longer, but it needs to be more efficient as well. And the way to make it more efficient is to have one gene coding for multiple protein products. That's that alternative gene splicing. We'll talk about that a little bit more again. But also, that means that we're going to need some smart way of taking this huge piece of DNA and packing it all up. Okay. I want you to picture going to Michael's or going to like any craft store, and you decide that you want to buy yarn, and you want like enough yarn to knit a sweater. And you go into a big bin in the back, and all of the yarn is just unraveled and put into a giant bin. What's going to happen when you go to pick pink out of the bin? It's tangled. You can't pull it out. It's all intertwined with other things. So eukaryotic genes are similar in that they're long linear pieces and that there's a whole bunch of different chromosomes, a whole bunch of different pieces of DNA. And so in order to package that up in a way that isn't going to get all tangled, we're going to use these little guys. These are histones. And these histones kind of come together and they make these little round globules. By the way, from the reading, what are those called? The histones with the DNA wrapped around it? Does start with N. So we've got these nucleosomes, and these nucleosomes coil to make hypercoiled chromosomes. We'll get into that tomorrow. But for now, what's important to know is this. If you want to make a sweater, are you going to be able to do it when your yarn is all coiled up? No, you got to uncoil it first. Histones are blocking transcription. RNA cannot bind to this. RNA polymerase. RNA can bind to the DNA, but it is not going to bind to that coiled up chromosome. Okay. Another fact that you need to understand is that different cells are going to be differentially expressed. What that is saying is basically that some genes are going to be turned off in some cells and some genes are going to be turned on. Example here, if you are a neuron, do you need to make the same enzymes as a liver cell? No, of course not. If you're a liver cell, do you need to make the same enzymes as a skin cell? Of course not. So in multicellular organisms, we need to allow for specialization to occur. And so chunks of the DNA need to essentially be turned off. Other chunks need to be turned on. So here's what I've done. I told you that the notes were super difficult. As you're reading it, a lot of information gets jumbled. So here's what we need to remember. We've got chromosomes. Those chromosomes need to be loosened up to allow for the DNA to be available. Transcription has to happen. Processing has to happen. The mRNA needs to get into the cytoplasm, get translated, get folded up into an active protein. Every one of those steps acts as a way to control gene expression. So if you don't need this protein, there's a whole bunch of ways that you can prevent it from being made. Either prevent it from being made or break it down. So those are the control points. Those are all the different ways that we can stop proteins from existing in a cell. And they are chromatin configurations, initiation of transcription. Those two are going to be the biggies. Those two are going to be the ones where you really focus your studying because it's a little bit tougher. The other four are just basic facts, okay? RNA processing, initiation of translation, protein processing and degradation, although there's a little bit of information here under the degradation of proteins. So, 6 different control points, 6 ways to control whether a protein is made. Are we feeling okay so far? What's that? I feel like I'm throwing a lot of information at you. So let's make sure to digest it if we need to. Okay. So I already talked to you about histones. These are the histone bodies, these are the histone tails, and they kind of shoot out sideways from the center of the nucleosome. DNA gets wrapped around and then moves on. Anything that condenses those histones, that attracts histones to each other more closely, like this, right? If you've got this close attraction, then you are not going to have transcription. Low transcription. If those histones are all loosey-goosey and spread out, that means the polymerase can bind and high transcription is possible. Okay? So anything that condenses will decrease transcription. Anything that loosens will increase transcription. Yeah, Ashley? No, they temporarily get moved out of the way. That's a good question. So these histones right here, if polymerase can bind, then these histones are going to be temporarily moved out of the way, then put back on. But you still have to have the RNA polymerase capable of binding, which means it at least needs to be semi-loose. This is not going to allow for that to happen. Great question. Great question. So it's not just transcribing these little pieces in the middle. Okay? All right. I thought the book did a pretty crappy job of explaining this, so let's go through it. If histones are loose, transcription occurs. Acetylation encourages that process. Acetylation loosens the histones and allows transcription to occur. On the flip side of that, deacetylation and methylation increases how compact they are. Okay, so deacetylation and methylation leads more to this type of structure. So acetylation would increase transcription. Deacetylation, which is just the removal of the acetyl groups that you did in acetylation. So deacetylation and methylation increases how compact they are. And that methylation can be kind of negated by phosphorylation. So it's not like phosphorylation increases transcription always. Only when there's methylation that's already decreasing it will you get this like negation of the activity of the methylation. Okay, recap there. Acetylation leads to increased transcription. Deacetylation and methylation decrease it. Okay, and not only can we modify the histones themselves, but we can go ahead and modify the DNA. The DNA itself can be modified using methylation, and this is where it gets super tricky because it's not really understood why this happens. In some areas it does increase how tightly packed in everything is, but not always, and it doesn't always lead to the turning off of a gene. But in general, just in general, it's DNA methylation. Areas of the genes that are methylated are less transcribed or completely turned off. Typically. Not always, but typically. I said yesterday, if you're sitting here thinking about getting into any kind of biology research or biotechnology or genetic engineering, to look into splicing. The other recommendation would be to look into epigenetics, because we really don't know how much there is to know. The more we're learning about epigenetics, it's been called the second genome. So your sequence of nucleotides definitely plays a role in what you end up looking like. But your epigenetics, the patterns of what's turned on, what's turned off, what genes are methylated, what histones are methylated, that also plays a huge role in how genes are expressed. So I'm going to throw my boys under the bus again here. You can tell a clear difference, despite the fact that they are genetic clones, between Bailey and Logan. They're genetic clones of each other. But the epigenetic markers on them are probably hella different, right? And that's going to be true for anyone. You can clone a cat. You can get this done. I don't know why you'd want that, because cats are freaking weird. But whatever. If you clone a cat thinking, oh, my little Fifi died, I want another Fifi. Guess what? Cat number 2 is not going to be like Fifi, because that cat is going to have different patterns of histone and DNA modification. So even though the nucleotides are identical, they're clones of each other, the same genes. How they're expressed is a little bit different. So Fifi number 2 might look different from Fifi number 1. And you can bet Fifi number 2 is going to act a lot different than Fifi number 1. So that is the idea of epigenetics, the idea that the DNA itself could have the same sequence, but how it's expressed can be different. Okay? So far, so good? 2 ways that this matters. First of all, for development, that's going to be huge. You start as one cell that has every single gene active, and eventually, as it divides, certain genes need to be turned off. So to make a liver cell, you have to make sure to turn off the right genes, and then every time that liver cell divides, it needs to have the same genes turned off. That's done via epigenetics. It's also an inheritance thing. So let's say I take up smoking because I'm bored, right? And then in 5 years, I'm like, well, that was a poor decision, and I quit. The smoking is going to play a role in what genes are expressed and my epigenetic markers. Then let's say 5 years after I quit smoking, I'm done. I haven't had a cigarette in 5 years. But I go and have a kid, which would suck because I don't want a kid in 10 years at all. At all. Anyway, guess what? The fact that I smoked at some point during my life is going to affect how that kid's genes are expressed. Not only my kid, but my kid's kid could be affected by that activity. What's an example from the book that you read over the summer of when epigenetics had a huge role? Do you guys remember? This group looks like they want to say it. No? That's straight up evolution, but good guess. And I'm pretty sure it talked about, right, how in times of famine, that kids grew up in famine, grew up and had kids who were more likely to be obese, and grandkids who were more likely to be obese. That doesn't make any sense if we just look at it from a genetic standpoint. Their genes haven't changed, but the epigenetics, what histones are modified, that's changed. And it's changed in a way that is heritable, and it is changed in a way that it does affect future generations. Okay. So before we even expose DNA to transcription, we can stop proteins from being made. Now, once the DNA is exposed, our second way of controlling eukaryotic gene expression is by preventing or promoting gene expression at the level of transcription. This is a very, very tough concept. This is the one I think that is the most tricky. So really brain in the game. Okay? Remember I lied to you the first time about transcription factors. I said they were in prokaryotes. They are not. Prokaryotic cells have polymerase binding directly to the DNA. Eukaryotic cells do have transcription factors, and there are going to be 2 types, general and specific. General are always needed. Specific depends on the gene. Okay, so here we're really going to focus on the specific ones. If you get rid of general transcription factors in your cell, all gene expression is gone, which is bad, so don't do that. But for specific ones, you might have some gene expression still able to happen, and other ones would be shut off. So, specific transcription factors can be activators or repressors. We're really going to focus here on the activators. To understand how they work, let's go ahead and take a look at a sample piece of DNA that we have here. We've already looked at exons and introns. We know that the promoter includes the ta-ta box. This is where the DNA is going to be binding. So the RNA polymerase, hello brain. The RNA polymerase binds to the promoter, transcribes to make the mRNA. That gets processed to make the finished mRNA, which only has the exons. But we have these other things upstream called the enhancer, and that's what we're going to really focus on, is this enhancer and these distal control elements. Distal just means far away, but the control elements are what is key. So our control elements are little sequences of DNA that are way upstream. It can be way, way far away, thousands of nucleotides away from the gene. And different combinations of these control elements are going to attract different activators. So each activator binds to a control element. That then can bind the general transcription factors to promote transcription. Raise your hand if that made sense. Absolutely. Each activator, which is a specific transcription factor, binds to a particular control element. The control elements are these little pieces of DNA, and they're variable, okay? So each activator binds to a control element. Based on which activators are present, you'll have different general transcription factors binding, which will eventually increase transcription. So the activators bind to the control elements, right? I'll draw these guys in dark blue. I guess that's just blue, whatever. And that's going to bind, okay, so these are our activators. And those are then going to bind general transcription factors, which are eventually going to allow for RNA polymerase to be attracted to this area and go. Technically, it attracts to the promoter, but there's some folding and whatnot, so we can almost just think of it like a big, giant complex. And that's where the RNA polymerase binds. Tons of information there to digest. Ashley?
So all the control elements need to have activator attached to it?
Yes. For it to act as a way to enhance transcription, each of the control elements that are part of it need their own activator.
Why are there 4 general?
I don't know, because I drew 4. Oh yeah, there's dozens. About a dozen that will attach. Okay. Britland, did you have a question?
Yeah, I thought, like you said, control elements were like at the very end, right?
Yes.
So then, I don't understand how, like, I
Oh,
don't
shoot.
understand how it increases transcription. 'Cause, like,
I didn't include the picture. So, okay, the question is, I'm repeating for the video. The question is how these that are way up here affect transcription. So really what it's doing is this is the gene, right? And then you have the promoter, and that is eventually where we're going to want to get the RNA polymerase to bind. These upstream, like distal control elements that are part of the enhancer, essentially what they're going to do is kind of attract the promoter here. So if we could attract the polymerase here, but then be able to fold back over and place that polymerase on the promoter so that then it binds here.
was that the picture you used that's like in this one?
Yeah, where it's like, it's got this and it's got this and it's a weird picture and I don't love, yeah.
Okay.
Does that help at all?
Yeah, because when it folds, it makes it go closer, right?
Exactly. It brings it into the closer proximity so that the polymerase is more likely to bind. Okay? And so the way that we control, because this isn't just about like getting the polymerase there, it's about getting the polymerase to only the genes that we want to be transcribed. And so the way that that happens is what is called combinatorial control. Essentially, each enhancer is just a different combination of control elements. And if that combination changes, then it's going to attract different enhancers, and different specific transcription factors, which is going to change which genes are activated. And there's actually a pretty limited number of control elements, so it's all about which combination are present to be able to activate the gene. Coordinate control, remember how we were looking at prokaryotes, and it's like the promoter, gene 1, gene 2, gene 3. So by starting transcription here, all 3 of these genes are transcribed. That is not the case in eukaryotes. That is for prokes only. And so related genes on eukaryotes might be on completely different chromosomes. It might have gene 1 of a metabolic pathway, gene 2, and gene 3 on 3 completely different chromosomes. And so the way that we're going to get them all activated simultaneously is because they are going to have the same, let's go with this color here, the same type of control elements. So the presence of those control elements means that all of them are going to be simultaneously activated. Ashley?
So the number of control elements and what they are affects like the rate of transcription?
No, not necessarily. But the presence of the control elements will affect the rate of transcription. More control elements, more specific transcription factors, stronger transcription. Okay, that's about as clear as mud, right? Really spend some time going through that to be solid there, because I think these are by far the most difficult. I said at the beginning that these 2 are really where eukaryotes get complex. And if you're curious about research, I mean, what I've just gone over with you guys isn't even the tip of the iceberg. It's a molecule of water at the top of the iceberg. There is so much here that we still don't know. Okay? The rest of this is a little bit easier, as in one line. RNA processing. You can change how mRNA transcripts are spliced together using alternative RNA splicing. So if you need more of this protein, or if you need more of this protein, that can be altered after it's been transcribed. You can change how well translation initiates. So you've got the mRNA, you've got the 5' cap and the poly A tail. If you block the ribosome from binding, then guess what? Translation doesn't occur. So you've got the mRNA, but you don't make protein. So that's one way to control it. If you break down the poly A tail, the ribosome isn't going to bind. If you then rebuild the poly A tail later, it will bind. So it's almost like an on-off switch there. Protein processing. Yeah, Ashley. Yep. You're just trying to process it a little bit. Yep.
Because I'm still confused about the
But
Like, does it make both of those, or can it make one or the other?
one or the other.
Okay.
For protein processing, you can control whether things get cleaved or broken down. You can control all of those level 4 protein structures. So you can have the primary, secondary, and tertiary structure, but it's not going to be functional until it hits that 4th level. So if the enzymes needed to make these things happen aren't there, then you've prevented the functional protein from being made, thereby controlling that aspect of the protein. Right? So let's say you're making an enzyme, you don't need the enzyme active yet because you have enough of whatever product it makes, then you just prevent it from hitting the 4th level. Sounds like a video game. Then when it's time that you need more of that enzyme, you just cleave it or add whatever chemical groups. All of a sudden it is functional and working. Okay, so let's say you've made a protein, or you've made the mRNA, and you don't need the protein product from that mRNA. Now you can straight up degrade it. This is the last way that you can control what proteins are present in a cell is by degradation. So you can degrade the mRNA. And so here's the mRNA. Here's the start codon. Here's the stop codon. And what these end pieces are called untranslated regions, UTRs. There are signals in the UTR that act as ticking time bombs. And maybe that mRNA will only last for minutes, and maybe it'll last for hours, and maybe it'll last for weeks. So if the signal here says, hey, don't degrade me for a while, I'll stay stable for weeks, then this gene can get translated millions or billions or trillions of times. If it's only active for seconds, then it's only going to be translated a couple of times. Okay. This is my favorite. You can actually mark proteins to be destructed by using this little tag right here. It's called ubiquitin, but the way that I remember this is ubiquitin. If ubiquitin, then you're tagged for destruction. You quit on me, you're out, right? So if this is the protein, we tag it with some ubiquitin. That's acting like a little chemical signal to say, send it to the proteasome. And the proteasome is like a little destruction complex that breaks everything down. So proteasomes are just looking for ubiquitinated. I think that's the right word. Anyway, ubiquitinated. Who cares? If the protein has ubiquitin on it, the proteasome is going to be like, bam, destruction, cleave everything, break it into wee little pieces, and voila. Okay? All right. I'm going to stop this.